Okay, I think we're ready. So welcome. He's well, I see Morgan said he's going to the hackathon today. He's going to the tower for that. And then Vidhi says that sounds interesting. So this is the student hackathon here. With the link in the chat. This is at Frontier Tower. This is a Global NeuroTech hackathon. It's presented by Frontier Tower. It's this 16th floor Nexus for Frontier Tower in San Francisco. Join us. And then, of course, Morgan is sort of the organizer there. He works for Neurotech X, so he's working on that. Next weekend, Neurotech X is hosting the student-led Global NeuroTech Hackathon with student teams from around the world coming together at Frontier Tower to build and collaborate together with a leaderboard. This hackathon is for student teams decided by their home institutions and not a public hackathon, so they're just recruiting people from the student clubs at McGill, Berkeley, Johns Hopkins, Stanford, and Austin and UC Davis. So it's looks like a really interesting event. Congratulations to Morgan for heading that up. Hopefully they do some really interesting good work. All right, did we have Vidhi? Did you have any updates?
College, reading a few papers, and yeah, exams will start in a few weeks. So like this is the lower calmer phase. So I'm just enjoying before I start studying a lot for that phase. But yeah,
That's good.
looking forward to just hug the summer.
Yeah,
Yeah.
it's gonna be good. Yeah. Yeah. So let's see. So the DevoWorm meeting this week. This was, I think, meeting 12 or 13 in the year, and it's on our YouTube channel for DevoWorm, not for orthogonal. And this was we're going through, I think, some residual uh, discussions about GSOC, and then we did some other things as well. So we had Shreyas, Barshan, Ashu, Karsh, Morgan, and myself to begin the meeting. We went through, Ashu had produced some data. She produced a new data set. It was based on something from Wormbase, so it was like a different way of like sussing out the daughter cells of different cells. So when cells divide, they divide in a binary fashion. They divide into two cells, at least in C. elegans, and you get these daughter cells. And the daughter cells, we can track all of the cells in C. elegans, and we have the nomenclatures for them. So whenever a cell divides with two daughter cells, and we know what those daughter cells are going to be, and even more interestingly, we know their relative position in the worm. So we know if it's going to divide in an anterior direction or a posterior direction or a left direction or a right direction. So we have these types of information available to us. And it unfolds the same way in every worm. So this is a very fortunate part of this model organism. And so this is just showing these different parts of this data set. So she built an identity map. She has a lookup table, some normalization rules, and so that's all going to get pushed to the GitHub repository. Then this is where we have some motifs from the connectome. So she was going through the developmental cells in this figure, or in this table. And then in this figure, she ran, in a notebook, she ran some network motifs and displayed them here. And so a network motif, if you're not familiar, is a small connected set of nodes in a network. So a lot of times we'll look at motifs like triangles, which you see here, where you have a triangle where three cells are connected fully, and you have this triangle. And a lot of networks will have triangles in them, or they'll have diamonds in them. And those motifs are, we usually count up the number of motifs, and it tells us something about how the network is connected. And so this is a method that people use In topological data analysis, they use it in community detection, and they use it in other techniques as well. Okay, and then this is her sort of flow chart for what we were looking at. And this is all in service of this method, hyper SAGNN. And this is vertex convolution, and then she's going to work through this in her proposal. So this is a nice contribution. We haven't gotten around to contributing data sets in a while. Like we've had some data sets in the past that we've committed to our GitHub repository. And they're usually built upon other shared data sets. So we usually have data sets we'd like to extract for our own purposes. So we have those in our GitHub repository. And so that's something that is well appreciated, I think. And hopefully people will use it outside of this project. All right, so then Utkarsh talked about his proposal, discovering developmental rules in the C. elegans connectome. This is using the Whitfleet connectome dataset, and then using the C302 platform, which is the platform that OpenWorm uses for their, the OpenWorm Foundation sponsor the construction of C302. And it's basically where you're simulating the connectome of the C. elegans, which is 302 neurons. And those 302 neurons, of course, each have their own biophysics, their own neurophysiology. So you're able to simulate that with C302 and then run whole simulations of the brain. And if you set the parameters in certain ways, you can simulate different behaviors. Then he wanted to use this in a developmental context, which we talked about in the meeting. And it's not the ideal tool because there are different things, a little bit different things happening in development than the adult. And C302 is based on the adult C. elegans, but it's the best tool we might have for that. So he tried that out, and his result was, it was acceptable. It wasn't optimal. You'd have to do some optimization of it, but this is his diagram of this experiment where he took the Whitfleet connectome, which is a connectome based on different larval configurations of synapses, and you have that for the different larval stages. Then you go to a C302 simulation, you plug that in, you then get neural activity from structure. And then you apply this other tool called CindyG, which is a graph version of Cindy, which is a platform for simulating neurophysiology and biophysics. So he's got that pipeline. So this is the first experiment, which is the Whitfleet connectome and C302 combined. This just shows like these different cells that were stimulated, and you end up with these voltage traces and calcium concentration traces, and you can look at that. We had also mentioned that some of the results he was getting out of his experiments were consistent with the Rich Club theory of C. elegans connectomes, which is that there are 302 cells in the connectome, but only a subset of those are really active. They're really highly connected, and they sort of coordinate the entire connectome. Whereas most of the connectome is not highly connected and isn't active all the time like this subset of Rich Club cells are. And I can't remember what the Rich Club cells are. I believe that some of these are Rich Club cells, but we just talked about that a little bit, and I don't know what the answer is there, but it's something you could do with this platform. So then we go on here, talking about structural versus functional development in C. elegans. So structural development in C. elegans is the physical wiring of the connectome. In other words, if you have cells and they have connections that are either electrical, which are the gap junctions, or they're synaptic, which means that there's a connection that's established between two cells. and they have an immediate connection. And whether or not they're active in any one behavior is immaterial here. What we're interested in is the structure of the connections between the cells. And so in the Whitfleet data set, or at least in the data that they've presented in their papers, they have these electron microscopy snapshots of C. elegans, which means they're very high resolution images of C. elegans. And from that, you can see those connections. You can actually enumerate those connections. In fact, this is the way we usually sort of enumerate connectomes. We look for different connections in microscopy images, and we isolate them and then establish that two cells are connected. So this is, and then of course with electron microscopy, you can make synapse counts. You can do, you can actually see the synapses at the end of axons, at the end of dendrites. You can actually count them up, and then have a count for each one cell sort of communicating with another cell, and you can look at changes in those in larval development. The other type of connectivity is functional development, or at least the other type of development is functional development. This is where you actually base your connectome on activity. And in neuroimaging and say like humans, we have that distinction between structural and functional networks. Structural networks being the what cells are connected, or in this case what voxels are connected, and then functional development is which ones are active at any given time. So that we have that in C. elegans as well, except we can isolate the cells, and we know from calcium imaging data, which is where they inject a tracer, and you can actually image calcium influx to the cells. And so kind of if a cell is active or not. You can then make a distinction of what cells are functional in what circuit at what point in a behavior. So, some of this, of course, is captured when we do optogenetics. We can stimulate cells. We can also look at cells that are active given that stimulation, and we can get a better handle on actual activity of these networks. So functional connections are sort of a subset of structural connections. So he's making that distinction. And so some of that is where we're doing the CinDG experiment. So this was on Whitfleet. So this is where he was able to do some other work with the neurophysiology, set up the simulation in CinDG, and then looked at it that way. And then he has these cubic spline fits, which are based on the synapse counts from Whitfleet, and was able to simulate this for different cells over time. And so then he does this experiment with weak CinD, where he was able to discover equations that described the connection between two cells, or at least the activity of cells. And this just kind of goes over that experiment. And that was really interesting work. We had a larger discussion. I can't remember what this discussion was about, but I guess it was about the experiments he was doing. And then finally, we had this paper, which is spatiotemporal segmentation of contraction waves in the extraembryonic membranes of the red flower beetle. So this was a paper that we've been kind of sitting on, but Susan Crawford Young, who's here, she sent me this paper and wanted me to go over it. So the idea is that in the embryo, you have these calcium waves that go through the embryo early in development, and it organizes the sort of cell differentiation, meaning that it organizes, and there are a whole bunch of waves like this in the embryo. They organize how cells differentiate, the order in which they differentiate, and oftentimes coordinate cells in groups so that they can form tissues, or sometimes it coordinates them to migrate in a certain direction or go to a certain place. So these contraction waves, and these are things that they've observed. There's a theory of differentiation waves that one of our collaborators in DevoWorm has been working on for a long time, for decades. And this was interesting to that work as well. So that's a theoretical sort of synthesis of a lot of these different waves that we see in embryonic development. And so they're using this model organism, the red flower beetle, that where they know that these waves exist, but they're trying to characterize their function, and so that's why this is an important paper. And they use this approach called particle image velocimetry, which is PIV for short, and that is where you can look at the movement of cells using cell tracking and then extrapolate out their velocity. So you can know how fast cells are moving and what direction they're moving in the embryo, and you can build little maps that show the arrows, and you can show basically the orientation and speed of every cell in the embryo if you're tracking it. And so you can build these maps where there are these, you see actually that sometimes the movement is coordinated, sometimes it's not, and a lot of times you get these shifts in direction, and those shifts are thought to be the products of these waves. So we can do this from different types of recordings, 2-dimensional and 3-dimensional, plus time. And this was a nice paper because it really gave it really kind of broke this problem down in a single model organism. They had these maps here where you see the sort of the results of this PIV method, where you have arrows for that are sort of like these velocity fields, and it just shows that the cells here are moving in this direction and then up in this direction. And this is leading to different morphogenetic events. So this is leading to different things happening in those regions of the embryo, setting up differentiation events and different type, maybe like the beginning of formation of tissues or other types of folds in the embryo. And so this is just going through a lot of these data. You can find a correspondence between the speed of these velocity fields and the sort of time of developmental events or the onset of developmental events. So here they're looking at the onset of a developmental event, and then this function, which is a characterization of speed of in this velocity field. This is kind of showing this in detail. So then, we can do other things with the waves. We can look at wave segmentation. We can look at different sort of temporal aspects of differentiation. And it's really interesting, some of the things he did here. It's very valuable data, I think, for thinking about these waves and seeing how you can decompose them in different ways. So yeah, this is just really going over a lot of this work. And you can then map this to the embryo and see different regions, different anatomical regions, and what's going on there at that point in development. So very interesting work. And then Susan had some words to say about it as well. Okay, so that was a DevoWorm meeting from this past week. So yeah, now at the beginning of next month, the beginning of May, we're going to try to start our open source meetings back up again. So this is going to be about an hour-long meeting on Fridays. The time is usually 12 noon Eastern time, which is in North America. So if you're in another part of the world, you just have to figure out what the time difference is. If you are interested in joining, we'll be posting in the Slack about when this is, and what the topic will be. So basically, it will be when we select our GSOC students. They will be presenting their projects during this time. We also welcome anyone to join us, and they can work on their own projects. If you're not successfully selected for GSOC, you can work on a project there as well. And it's and then we'll also have, and this is usually something that the mentors do. I do this a lot. Jesse does it sometimes. Sometimes Morgan joins in. But anyone's welcome to talk about different topics in open source. So we usually, they're usually somewhat obscure topics that are, something we'd like to talk about and kind of think about a little bit. We also talk about project management techniques, and making sure that we can give people some professional development opportunities. So to think about some of the things you might encounter in the workplace that are, you know, dilemmas, or sometimes younger people haven't thought about because they've never experienced it. So that's always good stuff. And so we'll be starting that up around the beginning of May. And hopefully, it'll be a good time this year. Last year was pretty good. I think we got a lot of interaction and a lot of productive time out of that. All right. Looks like Morgan switched to his laptop. Also, Sarrah's here. Welcome.
Thank you, everyone. I've been here for some while. I was listening to you, Vidhi, explaining. Yeah, good to be here.
Good.
Thanks.
Yeah. All right. Maybe we want to share anything today?
Not much. The past week, I've just been focusing, I've been going through the proposals. Also, some interesting stuff on. Are you all following the GSOC mentors thread by any chance?
I have. Yeah. There are a lot of things going on there. Yeah.
A lot of things happening there. I wake up every morning and while I'm going to work, I just go through the emails there. But yeah, it seems like, everything that's happening, it's kind of relatable for every org, right? It's the same things are happening to everyone. And there's no one has kind of a solution for it. Everybody just has their own techniques. And yeah, but interesting to see everyone's kind of approaches to it as well.
Yeah.
And yeah, mostly just reviewing our proposals. There's quite a few this year. So last week's just been that and work.
Yeah.
Basically.
So, how many proposals did you get for the large language models?
I kind of starred every that was both, so 7 and 8 both have starred and kept. So that's, the open source sustainability project has 14 proposals. Out of that, I think I'm not fully sure how many are for the project number 7, the Greenfield project, and how many are for the LLM one. I think I know 2 definitely that are for Greenfield so far. But yeah, another thing that I think we should really do next time for us is make sure that they all title their project name with the exact project title from the GSOC ideas list with the project number, because everybody has kind of come up with they've titled it innovatively, but it's kind of a pain to filter it through and then find everything correctly.
Yeah.
So yeah, but I would say 14 proposals for the open source sustainability project.
Okay.
Which is a good number.
I didn't count those, but I did count the Greenfield. I think there were 2 for Greenfield. I don't know why they chose
so the
Greenfield.
2 that I've already.
Yeah. I don't know why they said Greenfield. Meanfield would be better, actually, but whatever.
Yeah, I was a little confused. First, I thought that was a different orgs project, and then I read, and then I was like, "What was just named?" I knew you were submitting 2 projects this time. One for if you want to have any other approach, and one specifically for the Llama one. But I didn't see what it was named, and then I saw with all the proposals. And did Arnab give that name, or did you propose it?
I have no idea where they got it from, but
Probably some AI here too.
Yeah.
But yeah. So 14 is a large number.
Yeah. Yeah. It
But
is.
yeah.
Yeah, we had like I think 10 for DevoWorm, so that's a lot as
Oh,
well. So
compared to past years, yeah.
yeah, well, I mean, every year like we get our projects usually get pretty good interest. Like, and I don't know
Ah,
what other projects are getting, but like usually we're in like I mean a few more. Like I think last year we had like 7 for DevoWorm and like 7 for open source
open source
sustainability.
was similar,
Yeah.
yeah. Yeah. Yeah.
So
Yeah. But then still I would say it's doubled this year, right?
yeah,
It's
yeah.
like a quick
So did you
but
find?
still yeah, sorry, go ahead.
Oh, did you find the rubric useful that I sent out, or?
Yeah, I did. I'm filling in that. It's
All right.
good to kind of break it all down into individual components
Yeah.
to be able to instead of just. I did add a few more columns though, but I'll share it with you.
Okay.
Yeah. Yeah. But yeah, just that. Is Jess not coming? Yeah.
I don't know. He might come later. Usually comes later, but hopefully he'll maybe he'll say something about like some of the other things he's been working on. But
Yeah.
all right. Thank you, Sarrah.
Thank you.
So I did want to talk about this. This was a couple weeks ago. Of course, we had the embodied intelligence workshop, and this is a good time. I think there was a lot of interesting stuff at that workshop. A lot of uh, soft robotics, a lot of embodied robotics, and then of course you had other talks that focused on cognitive science and like computation and things like that. Um, and so this was one of the things they had this year were these uh, master classes, and they were basically experts in different areas who would talk about their um, different areas of expertise, and they'd put on a class, kind of giving you an introduction to something, and then it would be, on that topic. So one of the master classes was on reservoir computing, and if you're not familiar with reservoir computing, it's a sort of a newer technique. It first was proposed in around the year 2000. And it was this idea that you can do sort of, maybe, you could think of it as like an alternative to different types of neural networks, where you have a reservoir of nodes and you're putting together networks based on data that goes into this reservoir, and then it comes out the other end as sort of this uh, basically this encoding or this representation. And so that's kind of the idea behind reservoir computing. And it actually started as something that was made for analyzing problems that involved reservoirs, which is interesting. But it's soon evolved into this idea of reservoir computing, where you have different computational resources, and you're using, you're trying to optimize those resources. And then, of course, they have reservoir networks, which are it's something in network science that's taken off, where you use reservoir computing devices to build networks from data. So you have your data, which is your interaction matrix. You put it into this reservoir, which is a sort of a stochastic process, and it puts together this network that you can then use to analyze things. So you have this representation that comes out of it. So it's a pretty nice little method. I know they were thinking about this in OpenWorm at one time, how to use reservoir networks to characterize the connectome of C. elegans, which would be a nice little project because it's a very small network that you're trying to reconstruct. And you know, it's a computational tool that doesn't have a huge amount of overhead, so it's, you know, plausible to build something like that. So this is something from the Morphological Computation blog, and so this is talking about this PHRESCO competition, which is the first international physical reservoir computing competition. So this is Physical Reservoir Computing Competition, or PHRESCO, is the world's first competition dedicated to exploring the frontiers of physical reservoir computing. We invite researchers, students, and interdisciplinary teams to demonstrate how physical systems, from soft materials to chemical systems to biological tissues, or any complex physical structures, can be harnessed for computation, intelligence, and complex signal processing. So, like I said, reservoir computing started with actual reservoirs of liquids, and you know, they were using that as sort of the metaphor. But people are also interested in this sort of physical computing, which is where you have physical systems that are, you know, basically the computation machine. And you know, we talked about this with different types of chemicals, or you know, you could maybe include organoids in that, or slime molds, which we've talked about before. Any of those are sort of ways that you can do these computations using this physical reservoir structure. And so this is not just a contest, it's a collaborative research initiative and completely free. You know, just so you're aware, it's not like a money-making scheme. In this emerging field, your input will help define what counts as creativity, novelty, and significance. So they give a little bit about physical reservoir computing here, which is PHRESCO. It's a modern machine learning approach that uses nonlinear physical dynamics. So, you know, thinking about like a reservoir, like water flow, or laser dynamics, or in even soft robotic bodies. So you can use any kind of physical system as the reservoir and use those as computational resources. We are currently preparing a set of introductory videos to explain the underlying principles, but we're also preparing a few examples for inspiration. So they recommend reading this paper, Physical Reservoir Computing in Robotics, which looks at how soft bodies of robots can be used. However, PHRESCO goes way beyond that. The first ever physical reservoir computing work used a bucket of water. This is this paper, Pattern Recognition in a Bucket. This was Crisantha Fernando and Samsa Subjaka. A good overview is the book Reservoir Computing, Theory, Physical Implementations, and Applications, which provides a chapter on general reservoir computing, but also some physical reservoir computing. Finally, a look at Susan Stepney's Physical Reservoir Computing, a tutorial, which is at this link. So these are all like A-Life people. Susan Stepney and Crisantha Fernando are, you know, work in A-Life, artificial life. And, you know, this is consistent with this idea of sort of non-conventional computation. Okay, so that's, I mean, this is just kind of going over this work. And so then, you know, who should apply? Academic researchers exploring novel computational paradigms, students interested in unconventional computation, cross-disciplinary teams, which might include physicists, materials scientists, neuroscientists, and engineers, independent researchers and enthusiasts with a passion for experimental systems, then everyone who is fascinated by physical reservoir computing. So this is just kind of, you know, trying to be inclusive to all sorts of different fields, as this would involve not just conventional computer science. So this just goes through the criterion for this. This is a picture of, I guess this is just showing an example of an acceptable implementation of reservoir computing. So this is a very small example here. This is a crumpled piece of paper with dots on it. So these are fiducial markers for output extraction. There's another dot that's in green, so the output extraction are in blue. There's another marker in green for input. And so then you have the servo, which serves as input actuation, and then an Arduino for servo control, I guess, which does something with the input, and then it gives you outputs. So I'm not really sure the details here, but this is basically a reservoir computer. And, this is like, I guess, an acceptable submission. So you have to kind of read the papers and figure out how to build the computer that you want to build. As that there's a lot going on in different types of computation beyond neural networks, because we talk about like neural networks and large language models a lot in the group. We've talked about a lot of other kinds of computing in the past, but just as a reminder that there are all these other paradigms out there. Okay. So I don't know if Morgan's there.
Yes.
Yeah.
Yeah. I mean, I was just talking to some users of Final Spark, the organoid intelligence cloud platform. Basically they run a bioreactor farm in Switzerland that is, using organoids connected to microelectrode arrays. But the primary, at the demo days or what, I forget what they called it, user days, a lot of the applications were just reservoir computing approaches, and that was also the I forget if it was, I think it was Indiana, like one of the first organoid machine learning examples was also a reservoir computing, and if I would say, not a full criticism, but just like, this is something that you can do with non-living matter. and it's an interesting, so I think it speaks to, well, leaving aside what it says about organoid computing, it's still, it's still an interesting topic, and if that's one way to perhaps have a submission, like that's cool, and I think I know some people who should at least look at it, because they're definitely taking this reservoir approach. I just need to learn more about reservoir computing. What's its lineage, and what and then like what does it make sense to compare and analyze the reservoir computing approaches with,
right?
so that you can say more about what just so you can say more about what the system is doing. Explainable reservoir computing.
I think the problem is that it's not very interpretable. I mean, as you can imagine, because you have this big hidden pot of nodes, and you have hidden states in there, and you have to kind of extract things out of it. I've been doing, like I think I mentioned, I'm doing this agentive coding paper that where I've actually taken reservoir computing and then compared it with diffusion processes, like forward and reverse diffusion So that diffusion is a hot topic in like machine learning.
Yeah.
And like there's this, you can use those kind of as a comparison. So you have this sort of physical, you have this physical or quasi-physical process of some physical randomization, or so forward diffusion is where you're adding noise into an image, say, and then put it, doing a reservoir network, you might take an image and have it as input to the reservoir, and then it's trying to find the. You ever heard of self-organized maps?
Yeah.
Yeah. So I mean,
I met Conan.
Okay. Oh, good. Yeah. So I mean, it's kind of like that kind of process where you're taking a bunch of random nodes and you're kind of finding the pattern from the map. And in the reservoir, it's just like you have a reservoir with a finite number of nodes, and you're just kind of extracting the features out from there. And then in diffusion, you're doing forward diffusion where you're adding noise, and then that's helping you to recover things because you're generalizing the model. So as you add noise into the images, into that input, you're sort of saying, okay, this input looks like this in an idealized way, but then if I add noise, I'm actually able to see what it looks like partially occluded or with a partial mask. And then the reverse diffusion will recover images from that noise, so you can recover a wider range of images. If you're adding an input into a random network, you're able to recover patterns based on sort of those, that random network being rewired into a feature space. And so that's kind of the way that the comparison works. You could compare diffusion and reservoir networks and how those things yield an analysis of some sort of input data. But the difference, of course, there are a lot of differences. One, of course, is interpretability. And I guess in the case of diffusion, it's higher, and in reservoir computing, it's lower. There are also some computational differences. I think reservoir computing requires more computational resources. I'll have to pull up that. I generated a table on this. Yeah, just from my own curiosity, I want to know what the differences were. It's interesting. But yeah, you can. That's, I think, the most direct comparison.
Interesting.
Yeah.
Okay. I mean, I would really love to see that if you don't mind sharing, because this is such a widely used, or it's practically the only technique used in organoid computing.
Yeah.
I need to have more a better understanding in order to kind of shift the conversation, and this group that's meeting this summer to somewhat more focused on, focused on the whole range of OI applications, or kind of like issues. So that's going to include actually growing them and things like that. But this is definitely one of the topics that needs to be a part of this workshop, which is which it was nice to see that the organizer is definitely coming from he's definitely more of an engineer, but he's coming with enough ML background that he's got some really I think good questions about understanding what the reservoir computing approach is saying. When the way they're using it, they want to say that the tissue is we want to say that the tissue is doing work.
Yeah.
and that's the bit that is he's got some interesting ways to kind of probe that. So it'd be great to put that in the context of kind of some language that I understand a little better.
Yeah.
Or at least that's those comparisons. That sounds interesting.
Yeah. So the tissue is doing thermodynamic work, It's just the computational work is what you're saying. Like,
right.
yeah.
Well, I mean, like again, like when cortical labs, which is it's like this is kind of I'm holding them to an unfair bar, perhaps, but when cortical labs is calling this a biological computer, it's suggesting that they're passing data to neurons and that the neurons are processing them. And, and then they're passing the data back, or at least that would be a computer metaphor.
Yeah.
What you're describing is I would just say is more accurate, and is as we know, can be done by non-living systems.
Yeah. There's a metaphor there. And then there's this whole aspect of physical systems compute, which we did a whole bunch of work. We did a whole series of like features on in this meeting about 2 years ago now. Remember the physical computing thing? That is kind of thinking about that in a little bit different way. So, I mean, the idea there we were talking about like, is the brain a computer? Is it like is there physical computing that's comparable to to sort of digital computing? With reservoir networks, you really do have like a physical computer where you have things that maybe aren't bits, like they don't originate as bits. Maybe there are things that are changes in state, like in physical systems, you have phase transitions, right? And phase transitions are this rapid reorganization of the sum of molecules in, say, like a liquid going from water to a solid, or a liquid to a solid. And so in that case, you have this mass reorganization of things. You can look at that phase transition as, say, like maybe hardness of state of the medium, and you have this shift. It's kind of like a sigmoidal, where you go from like a liquid and then all of a sudden to a solid, and it's very sharp. It's not a step function, but it's kind of like a sigmoid, or it could be a step function. Now, what we'll do what we have to do to convert that to bits is to say everything in either side of this phase transition is either a 0 or a 1. So if it goes from a 0, which is a liquid, to a 1, which is a solid, that's a bit that's flipped. Or if we go the other way, it's a bit that's flipped. But that process is kind of obscured. We can't. I mean, we could. I guess we could capture that with a digital computer, but we'd always have to state that in bits. We'd always have to convert that to like a binary state, or maybe a ternary state if we're lucky. But that doesn't describe a lot of that process. So there's a lot of process that gets eschewed, and a physicist would be horrified because it's like you're obscuring this whole process. It's interesting. The same thing with organoids. You have these graded signals, and you have a bunch of stuff in parallel, and you have networks where the networks are these cells that are connected, and then the cells are in a state, right? And the state is just really kind of abstracted from a lot of complex, interesting biophysics going on. And that's kind of maybe the problem here, is that you have this, you say computational work. It's like, well, computation, what level of computation? What do we mean by computation in the first place? That whole literature on like unconventional computing. I can't remember how they're dealing with those things. I mean, it'd be kind of interesting to do a deep dive into that, to see how they frame those kinds of questions.
Absolutely, right? Absolutely. It reminds me sorry, I'm still on the way to the power, but like there's a company based in LA. it's funny because the CEO is like a super fan of Carl Friston.
Oh yeah.
But he's and they finally got aired up, you know. But blanking on the name of the company, but it's Exo something, and I think part of it is coming from. I was just gonna say, like, this is like a former quantum computing person who has started this compute company, not quantum, but it's got some sort of thermodynamic. It's like exo something.
Yeah.
And I wonder if it's like somehow trying to use a similar, you know, extra physical computation process. Is billing it as like a super efficient, you know, not neuromorphic, but yeah, I'll find it when I get to the tower.
All right, yeah, that's good. Yeah. So yeah, there are like, even if you go back to Feynman, he talked about a little bit about like physical computing and how you compute physics. And this is classical physics, not necessarily quantum physics, because quantum computing, of course, is its own paradigm. We don't want to borrow from that for classical processes. So, you know, it's kind of like, how do we characterize, you know, physical computing in that way? So yeah, I don't know. I mean, you have to look into that in more detail. And, it'd be an interesting set of things. I think I actually mentioned some of Feynman's work on it back when we did that physical computation series of features like 2 years ago. We have a post on our Medium about it if people are interested in going back and reviewing that. But yeah, just a series of discussions we had on different papers, different things. It ranged from philosophy to physics to computer science to neuroscience. And it's just kind of thinking through all these questions that were interesting to our group, but also more inclusive to different debates in the scientific literature. Okay, thank you, Morgan, for that discussion. So this is something else. I think Morgan posted this in the Slack. And this is a paper from the, there's this, it's on scientific replicability. Lead author on this paper is Brian Nosek. And so this article is titled Investigating the Replicability of the Social and Behavioral Sciences. It's very ambitious because there's a lot of social and behavioral science out there. But basically, they're doing a meta-analysis on the field, and they have this interest in replication and replicability. And so when we say replicability, what are we referring to? So basically, in science, one of the things that we try to do when we do experiments is we would like to, first of all, have a controlled experiment where we have a hypothesis, and then we set our experiment up so that we can test the hypothesis without too many confounds. So the idea being is that when we have a hypothesis about, like, say, if I move my mouse across the screen, you know, I'm doing something with my hand, there's motor control involved. And if I perturb that motor control in different ways, for example, if I have a distractor light in the periphery of my visual system, it will slow down my ability to move the cursor across the screen because I'm distracted visually by that thing, and it disrupts my sensory motor integration, and so my movement is slower. And you could, have a control where people aren't distracted by the light, experimental condition where people are distracted by the light, and then have different levels of distraction, so that one is obviously going to have an effect, and maybe one has a minimal effect. And you predict all this in advance. You design the experiments so that you can test the relationship in a very isolated way, and you try to eliminate confounds, which are things that could be other potential causal mechanisms or effects, and you try to minimize the interactions of different variables. So you just want to have the two variables, or that you're testing. So you have a very clean. If you have a very clean experimental design, first of all, your effect will pop out, and you'll be able to see the effect. You can use statistical testing to confirm that effect. And then, of course, you have an effect size, which depends on the number of people who are in your experiment. And then, of course, you have this idea of replication. So if I design a good experiment and I have this result, I can, do all sorts of different statistical tests. I can even do causal modeling and say, this is, the effect and it's real. Another way to know that it's real is to replicate that result. To do like 10 experiments and have them all basically point in the same direction. Or have 10 different groups do the same experiment. And why do you want to use different groups? Because different groups will implement this slightly differently. They might use different variations on the light. They might use different variations on the cursor speed and so forth. So you have, if the effect holds up across these different variations, then you know that the result is robust. So that's the idea behind replication. We'd like to see replication also because if we're trying to apply our findings to, applications, like if we want to build this into a computing paradigm, like a user interface, we want this to be something that is robust. So if we say, we want to build this result into a GUI, we want to make sure that this isn't just some result that's not very well supported and is maybe the case only about a quarter of the time, because that would be, it would result in a bad product. So replication has a lot of uses in, in the behavioral sciences, but also in other areas where we do experiments. And, the thinking is, of course, is that physical systems or physics, you can replicate things very easily, or maybe in chemistry, because those are physical systems, and we have a very high degree of characterization of those systems. And quite frankly, they're not as complex as social systems, or so the thinking goes. So a lot of people who do biological experiments and social science experiments will say that, I can use experimental techniques, but they're not going to be as replicable as those in physics and chemistry, simply because it's harder for me to isolate the different variables involved, not get interactions, and not get confounds. Because, in biology and in social science, you have sometimes you have uncharacterized variables, sometimes you have a lot of things that are interacting, and it's just really hard to isolate those things, and then also make that experiment useful for understanding the system as a whole. So we can do experiments in biology and social science that are very contrived, but they have very low, what we call ecological validity, or the ability to transfer those results to reality. What does that mean in reality? What does that mean in like a real social setting, or a real psychological setting, or a real biological setting? And a lot of times our experiments are just that. They're sort of artifacts of that don't have a lot of application outside of their own domain. So this is kind of where we sit with this. So the idea is that replication, we kind of assume that replication is the gold standard. In the social sciences, because of the factors that I mentioned, it's hard to get experiments to replicate. So this has led to something that they call the replication crisis, which is that you can do all these experiments and get these results. And if you go back to the literature to try to reproduce a lot of experiments, sometimes a lot of famous experiments, you can't replicate them. And this is a thing that is sort of the basis for this science reform movement that you might see. Brian Nosek, of course, is very interested in this. There are other people as well, which is that we need to improve science by finding better ways to do science, finding better ways to standardize science, because we want to have more replicable social science, more replicable biological science. Basically, where we can't replicate something, we should try to rethink how we do experiments, how we do data analysis so that we can make things more replicable. And of course, then there's this other school that says, well, we really can't do that. We just have to go with what we have. And, that's just kind of our heuristic. And that's the best we can do. And I wouldn't worry about it too much. So, there it is a debate. So when they present this stuff, it's interesting and it's, I think, useful for understanding where we stand in the science, in the social and biological sciences. But it's also very much, maybe we can't expect to use experimental methods and really expect to have highly replicable results. Maybe the best we can do is like quasi-experimental methods that give us indicators of what things are, effects in social science. If we have an intervention, for example, there's certain effects that we should expect, and it's not going to be very easy to replicate those across different labs or different contexts. So that's kind of where we are with this. So let's go over the abstract of this and then a little bit into the paper. So as meta science, what they're doing is they're taking a bunch of different results from different papers that have been published, and they're reevaluating those, and they want to understand kind of as a whole where we sit, how replicable are claims that get published. And sometimes they're very famous results, and sometimes we base a lot of our theories on them, or a lot of popular sort of science, popular ideas about how the brain works, for example, on them. So, there have been a lot of examples in psychology where you've had famous results that have everyone has kind of hung their hat on, and then they turned out not to be replicable. And so this is a little bit of a ding on some of the cognitive science techniques that have come out of the cognitive revolution. So in any case, let's get into the abstract. Pursuing replicability or independent evidence for previous claims. So you do an experiment and you publish your results and you observe the effects there. And then some other group tries to do the experiment again, and they either get an effect, and maybe it's of a different size because sometimes you don't get the same number of subjects. Sometimes those subjects exhibit variability. Sometimes your environment is a little bit variable. And so you can't expect the results to always be exactly the same, but you should get them in the same direction. If they get a positive result, you should also get a positive result. And you should be able to do this in any kind of setting. So like, again, if I want to confirm some finding, I just simply run the experiment for myself and I get that result. And as we know, we can do that. Actually, we have a version of this in computer science where if you run things in a container, the whole concept of that is that I will write a program, execute it on my machine, and then if you execute the program on your machine, you might get a different result because your machine is different parameters, has a different processor, whatever. But if I put it in a container, I can always run the program exactly the same way, and get the same results. So there is a version of this in computer science. Unfortunately, we can't containerize social and behavioral science experiments or biological experiments. So that's something to keep in mind. So this allows us to have generalizability, which in machine learning we worry about with training models. But in a lot of these experiments, we want to design experiments so that the results are generalizable to any kind of replication and eventually even to application domains. So here we attempted replications of 274 claims of positive results from 164 quantitative papers published from 2009 to 2018 in 54 journals in the social and behavioral sciences. So a wide range of different findings published recently. So this is, 2009 to 2018 is well within this sort of science reform era. So this is like, if you go back to like the 80s and 90s or in the 70s, you can go back there and find a lot of experiments where people were totally not thinking about this. They were very not thinking about replication too much. Sometimes they were, but a lot of times they weren't. And you might say, I'll take some experiments that were published back then and reinvestigate them, and lo and behold, they don't replicate the way I expect. Well, that's maybe unfortunate, but it's not something that where people are actively engaged in this conversation. But from 2009 to 2018, this conversation was going on. So, you might say, well, people were, just trying to run their experiments and live their lives. But the idea being is that, people are aware of this problem and it still persists. So, and I will say that the Nosek group and some of these other groups, they have a specific sort of agenda for this. It's not like they're picking on people, but they do have this idea that, science needs to be done a certain way. So this is, and they want to sort of advance their own research in science reform. So there is that aspect of it. So, I'm not saying that like people are just being intentionally dense about science reform. It's just that that's where we are in this debate. So they had a pretty good sample of different studies to look at. Replications were high powered on average to detect the original effect size, with a median of 99.6%, used original materials when relevant and available, and were peer-reviewed in advance through a standardized internal protocol. Replication showed statistically significant results in the original pattern for 154 of 274 claims, which is around 55.1% of studies surveyed. So this is, maybe not great. It's over half, but it's only about a little over half. So this means that, if you have experiments that you take out of literature at random, only a little bit more than half of them replicate. And that's, again, this is using sort of the, as close as we can get to the original experimental design. So, that is kind of disappointing maybe. And for 80.8% of 164 papers, which I don't know how they get the 80.8 of a paper, 0.8 of a paper, but I think this is just a weighting scheme, weighted from replicating multiple claims per paper. So this is where, they make more than one claim from, or maybe more than one experiment in a paper, and you're kind of looking at this in its totality. So we're really kind of when we do that, we actually get a little bit below 50%. So actually, if you think about this as an at-chance endeavor, like if we ran 1,000 experiments and we got 50% of them that replicated, that's kind of at chance. That's not, but that's one way to think about it. There are probably reasons why this exists. I don't think people are doing a poor job of experimental design, but they never really kind of get into that in these papers. They always kind of just show that there's this lack of replication. And, the implication is always that there's a problem in science. I think probably a lot of this is going to be like psychology or something psychology adjacent. But maybe it's just, again, the complexity and the nature of social and behavioral sciences, that it's very complex and it's hard to kind of isolate phenomena in a way that sort of like physics. Okay, so we observe modest variation in replication rates across disciplines, and depending on the discipline, it ranged from 42.5% to 63.1%. So maybe some disciplines it's really hard to replicate. In others, it's maybe easier to replicate. But really, you don't get anywhere near 100%, and you don't necessarily get anything near 0% either. So, and then some estimates though had a high uncertainty. So again, we're making an estimate about this based on this sample of things, and it's, I guess, a representative sample. But we're hovering around 50%, kind of around that area. The median Pearson's R effect size was 0.25 for original studies and 0.10 for replication studies, which is, of course, there are replications in the literature, and of course we expect the replications, maybe from what we're saying here, to not be as robust as the original studies. So again, we're finding this, we're kind of confirming this idea that the original studies are not great in terms of replication, but the replications aren't great either for obvious reasons. 13 methods for evaluating replication success provided estimates ranging from 28.6% to 74.8%, which is a median of 49.3%. Some decline in effect size and significance is expected based on power to detect original effects and regression to the mean, because we replicated only positive results. So you might be able to replicate a negative result, but that's not that hard to do. I mean, you do a poor job of setting up the experiment, and I got a negative result too. You don't even have to run the experiment to get a negative result, I guess, technically. But so having the positive result, that's sort of the thing that requires you to have good experimental design. A negative result doesn't necessarily require that. We observe that challenges for replicability extend across social and behavioral sciences, illustrating the importance of identifying conditions that promote or inhibit replicability. And again, this is assuming that you can do this in the same way that we kind of have a mental model for. So, again, why do we care about replication? Well, one of the aims of science is to discover regularities, statistical regularities, and sort of structural regularities. And one way to do that is to say, if I run an experiment and I get a positive result, if I rerun that experiment, I should get a similar result, and I should always get that result if this is a phenomena that's true or that's real. If it's not real, I might get a positive result once and then a negative result again, and maybe I keep getting a negative result, and then maybe I get another positive result. And this is not trivial, because basically when we do when we assess statistical significance, that's essentially what we're doing. If we had, say, 20 experiments, we should expect, if we have, say, like a 95% percentile criterion, that we should expect 19 of those experiments to be positive, to be successful, to show the effect, and one experiment that does not. So depending on our significance criterion, we should expect out of those 20 experiments a certain proportion of those to replicate. I mean, that's essentially what we're saying when we do statistical significance. We should say for 20 observations, vast majority of them should be in this direction, And there may be some in the other direction, but the reason why we have the significance criterion is because we want to say that, like, for the most part, this is true. There are always exceptions. So, people have talked about statistical significance before. There's this whole side debate about, like, the value of looking at statistical significance, and how people do things like p-hacking to try to, like, make something statistically significant that otherwise wouldn't be. So there are all sorts of problems there as well. But the idea is that we want to discover these regularities. So we want to be able to replicate our results so that every time we do an investigation, we can get similar results. And it should be reliable. And especially if we have something that is an application, we want to make sure that it's a repeatable result. And so then, they talk about this replication crisis and how a lot of published work will claim, well, it doesn't claim replication. It's just that they're published as positive results. And a lot of times people don't question that. And they kind of run with the result. And so this is a problem that we have, especially in things like cancer biology, where the stakes are pretty high, right? We need to be, if we have a clinical trial there, it's actually pretty important that we can replicate it. And a lot of times we can't, and I have thoughts about that, where it's like we're dealing with very complex systems, and in fact, with cancer, it's very it's highly variable in terms of how you observe these things, and so there are a lot of things there that kind of work against really good experimental results. But anyways, so they get into this. Popular explanations for the low observed replication success rates include underreporting of negative or inconclusive evidence for claims, high sampling error from small samples, and measurement error due to unreliable measures, coupled with a statistical threshold that serves as a publication filter. So there's this idea that, like, sometimes when we, to get things published, we need to achieve a certain statistical threshold of a p-value. And so if we can't get to that, sometimes people will try to get to that using, less than rigorous methods to get there. And this is something that is maybe a problem because a lot of publications won't publish negative results. And if they don't publish negative results, of course, the people don't get published. And there's a whole incentive structure that is immobile in this case. So, this is where you have to publish positive results. You can't publish negative results. So there's pressure to get positive results, even if it means sacrificing replicability, or just simply doing something more nefarious, like excluding samples that don't meet the statistical threshold. So that's kind of one of the points there. Low rigor and quality control in research, design, and measurement. So if your experimental design is poor, or you don't control for all of your confounds, and questionable research practices that inflate the likelihood of obtaining positive outcomes. These factors are compounded by a research culture that rewards novel and interesting findings and discourages error correction. Again, these are just these incentives that are misaligned with the ideals that we would like. Replication failures are not necessarily due to the original findings having low credibility. Low replication rates can also be due to false negatives, poorly designed replications, selecting only positive results for replication, and differences between original and replication studies that are initially perceived as unimportant. More broadly, there are conceptual challenges in deciding what a replication of a previous finding is and how to assess whether a replication attempt succeeded. So we sometimes, even just using like meta-science, maybe meta-science, the problem is meta-science. Maybe the problem isn't in the literature. But that's something that is a little bit more, that maybe requires a meta-science. But I think really, this is just kind of an interesting kind of a way to think about our results that we read. Whenever we read a result, is this something that is, can we trust this result as replicable, or is this a one-off result that is interesting, but it's not going to, we really have to think through this problem some more. Interestingly, I know there's a lot of people who critique benchmarking in computer science. So benchmarking kind of suffer from the similar kind of problems we see here, where you have two programs and you benchmark their performance, and a lot of times people are trying to optimize performance down to like a couple decimal places. And how effective is that really? I mean, you can game those kind of statistics pretty easily. And so, sometimes people will, want to show that their model is better performing than another model. So there's a temptation there to sort of game the system for your model. There are a lot of challenges in benchmarking as well, and I haven't gone and found a good critique of this, but maybe I'll do that next time. So they go through this study here. This is table 1, papers and claims selected by replication attempts by discipline. So they cover business, economics, education, political science, psychology, sociology, and then the total at the end. So we have claims selected and replications attempted. So they've selected a number of papers with claims, and then they have that number there. So they're sampling, I think in psychology they have 950, in education they have 445, and there's a range of number between that. Papers eligible for replication means, which papers can we actually replicate the claims with. So that's a subset of those. Papers with multiple claims, they're a smaller number with that have multiple claims in them or testing multiple hypotheses. And then papers with a single claim, which is the balance. So the papers eligible for replication, those are the ones that will be the replications that they attempt. So not all of the claims can be replicated, or if they don't think they can, or they won't. So they do a subset of these. And I don't know what the criterion for deciding that was, but that's, that is up to the team that did the paper. So they had papers with replication started, papers with replication attempts completed, total replication attempts of claims, so that's a smaller subset still, and then unique claims with replication attempts. So this is how they kind of divided this out. They took a bunch of papers from different fields. They examined their claims. They then took each claim. Actually, they took each paper, and they then looked at the claims, and they figured out which ones they could replicate, and then they did the replications, and they ended up with a much smaller subset of replication attempts completed. So this is, again, where we have a bunch of things in the literature, we kind of winnow it down to things that are sort of low-hanging fruit or easy to actually do in an environment. So we're getting like the kind of the simplest experiments, or the most important experiments. And so there is this selection bias here to be aware of. That is, we're not testing every claim, we're testing a subset of claims. And so I mean, there might be something to say about that, where you're kind of testing the things maybe that are the least replicable, or maybe they're the most replicable. It's not really clear. So that's basically what we have here. So you're making, and if you look at the numbers, actually, I don't think there's a huge sort of bias towards any one discipline. I think, I think psychology, it's a little bit more replicable, or maybe you had more attempts in psychology than in other areas. So you had papers with replication attempts completed, 58 in psychology. You had started with 950 papers with claims. In education, you have 13 versus 58, and for education, you had 445 papers. So there's a little bias, I think, towards psychology, and because the team is, I mean, Brian Nosek's a psychologist, and the team probably knows psychology better. This is probably why you have this bias. So, again, this is not, like, the experiments are behavioral experiments, so they're not going to be wildly different from the typical psychology experiment. So that's kind of why, just something to be aware of here. Okay. So for table 1, representativeness across disciplines was maintained during identification and extraction of claims because we use random sampling strategies. Most of the change in representativeness occurred owing to the non-random process of selecting and starting a replication study. So education and political science decreased in relative proportion of the sample, and psychology increased. Okay, that's, again, what I just talked about. Compared with the original sample of papers, the proportion of completed attempts of unique claims was within 3.5% for economics, education, political science, and sociology, and more notable variation was observed for business and psychology. Representativeness of replication attempts was relatively steady by year compared with the sample papers. We selected papers and attempted replications in 2 phases, with 139 of the completed replications occurring from papers selected during the first phase and 25 from papers selected during the second phase. Replication success rates were similar between the first and the second phase. Okay, this then is figure 1, which is the replication success rates across 13 binary assessments for papers. So what is this? This is where we have binary success measures and for different things, and then proportion successfully replicated according to measure percent. So these are the different sort of ways in which I guess things are assessed. So you have this measure of success. You have meta-analysis at the top, which is where, I guess there are these different types of mechanisms or different types of measures that they're binning things into when they're looking at how replicable those measures are. So meta-analysis comes out on top. And then you get small telescopes, Bayesian meta-analysis, replication effect and prediction interval, subjective interpretation, replication Bayes factor, then skeptical p-value is down the list, sum of p-values is down the list, correspondence test is right with the p-values, Bayes factor is down below, and then the original effect and replication confidence intervals. So the confidence intervals are at the bottom along with Bayes factor. So I'm not quite familiar with these success measurements, but it's interesting that sometimes Bayes and Bayesian stuff comes out on top or near the top, and Bayes factor comes out low, confidence interval comes out low, and then correspondence tests, which are usually pretty weak statistical relationships, come out with p-value measurements. So I'm not an expert at in these different measurement techniques, but it's kind of interesting how those are distributed. Okay, so they talk about this a little bit more. This problem highlights a challenging problem, or this project highlights a challenging problem. Replication success metrics can be applied only to methods that meet their assumptions. So again, sometimes people use metrics where the assumptions aren't met by the data. So this is something that, again, is maybe a problem in replication, is that people use different metrics or they use metrics incorrectly. And that can always be, because metrics always have assumptions built in. So this is a problem, which you would think would be corrected in peer review, but oftentimes is not. People will give justifications for using a metric that maybe they shouldn't be doing. Okay. And so in figure 1, we present 13 replication success metrics. Some are necessarily binary assessments of replication success or failure. Others are simplified to provide a binary assessment for comparison. Okay, across metrics, replication success rates range from 28.6% to 74.8%, with a median of 49.3%. So that number, again, 49.3%. The observed variation highlights the impact of different assumptions underlying each approach. For example, the highest estimate of 74.8% for the meta-analytic combination of the original and replication evidence provides strong evidence of success because it includes original studies that provided evidence of success, and these tests are not independent of the original evidence. Variation in the observed replication rates are affected by both the assumptions of the binary assessment and the subsample for which the metric could be used. Figure 2 presents correlations between each pair of binary assessments for replication outcomes, which both methods can be applied. So that's this figure here. And then this is where we have these different metrics kind of compared to one another. And then we have this distribution of all 144 correlations. So this is just kind of thinking about these correlations between different metrics and how these are distributed. So, let's see. Correlation values are to the right of the diagonal over here. And so, what does he say about that? Figure 2 presents correlations. Spearman correlations were positive and relatively high, with some exceptions. For example, analysts almost always interpreted success on the basis of statistical significance, as reflected by a correlation of 0.98, whereas measures that use confidence or prediction intervals show relatively strong correlations with each other. And so this is where, again, we have this different types of measurements having, sometimes they lead to high replication, sometimes low replication, sometimes specific types of measures are interchangeable. But also, if you're using other types of measurements, they aren't as consistent. So sometimes, you'll have a replication where you use, say, like a confidence interval, and another one where you use a Bayesian method, and you're going to have a different result. Is that due to the experimental design? Maybe not. Should we do that kind of test? Perhaps, because sometimes it can tell us about how robust a result is. And so, again, this discrepancy in different methods, using different methods for the different studies, is partly because they treated replication outcomes both smaller and larger than the original outcomes as failures to replicate. So there is this aspect of statistical testing and their effect on replication. Okay. So the discussion here is about half the findings from a sample of social and behavioral science papers published between 2009 and 2018 replicated successfully with variation and success estimates across 13 binary assessments and effect size comparisons. Variation in replicability across disciplines was modest, with replication rates between 42.5 and 49% for fields that had more than 20 replications. These findings are consistent with the cumulative evidence across systematic replications, and they illustrate that there is substantial uncertainty in estimating replicability. Okay, so there are some conceptual, methodological, and inferential challenges to assessing replicability. Conceptually, it can be challenging to attempt a replication. Strictly speaking, there is no such thing as an exact replication. Of course, we want to get it as close as possible, but we can't always replicate the exact conditions under which the data were collected. There are always differences in units, treatments, observations, and settings. Researchers must make decisions about how to conduct a good faith replication on an original claim. So, for example, should a present-day replication of a 2009 U.S. study of political behavior that used President Obama as a stimulus use Obama again, or the current U.S. president, or use the leader of the participant's nation? So, if you study people in another country, are they going to respond the same way as the people within the country where the leader is from? So, this again is like something that you do a study and it's interesting for what you want to find, but then it might be very highly contextual and replicating that study may not hold. So, but this is interesting because a lot of times we will take a study like this and extrapolate out the effect. So you might say something of, does this result say something about the context of the United States and President Obama, or does it say something about more generally, electorates around the world and their respective leaders? It may be very constrained to the U.S. context. It may not. So this is a problem that you have with studies when they're set up and studies when they're replicated. Another thing we have to worry about is as to whether or not the theoretical commitments are correct. So sometimes we have a theory, and this is something that the theoretical commitment might be wrong, where you have sort of you interpret your result in a certain theory, and of course it's the wrong theory to interpret it with. So then methodologically it can be challenging to determine how to measure replication success. So again, we have these measures, these metrics that they use, but again, you can't use a single metric universally, and a lot of times it depends on the measurements that the original studies used, and there are a lot of problems there. And then inferentially, it can be challenging to determine whether original and replication studies produce the same outcomes. Each of the replication assessment criteria has strengths and weaknesses and may be based on different assumptions. For example, metrics combine original and replication evidence that are not independent tests and have relatively low sampling error. These tended to suggest the highest success rates. So again, tests that are not these tended to suggest the highest success rates. They might be useful only when it is safe to assume no selection or publication bias, and the emphasis is on cumulative evidence. So again, different types of studies may be more amenable to replication, and different types of ways to study replication are only useful in certain contexts. So you don't want to assume that studies were free of selection or publication bias when they were, and you don't want to select a study that has selection or publication bias. Okay, in summary, the question, did it replicate, can be difficult to answer. Fortunately, the answer for any given study does not matter much in the long run. Research is conducted on a study-by-study basis. Replicability is established via a cumulative body of evidence. So sometimes when we have a hypothesis or a theory, it's not so much that it's whether it's replicable or not. It's that if there are 100 studies in a certain area and they all yield different results, we can kind of figure out what the right answer is by looking at the accumulation of results. In other words, if a study is published and it turns out that it's not true, that the result is maybe a one-off, it doesn't really hold true theoretically, then we should be able to detect that by looking at 100 different studies and looking at the direction in which they point. And if a study is pointing in the other direction, then maybe we just are skeptical of it and we don't need to worry about replication because we can kind of figure out the correct direction of what these findings suggest. Okay, I think that's enough on that paper. I don't know if Morgan had anything to say about that since he posted it in the chat. Be interesting to hear his feedback on it.
Yeah, I mean, obviously, it's super important to talk about these issues. Brian and the center there has definitely been a big part of, I'd say, helping move that conversation forward in productive ways. And yeah, so it's I'm still greeting students and things right now. But yeah, no major comments on it, but just so valuable and so just nice to see how I forget they I don't know if you've ever seen a podcast, Decoding the Gurus.
No, I've not seen it.
I highly recommend. I mean, it's some people who it's two, I think one's an anthropologist and one's a psychologist, but a methods psychologist,
Yeah.
And anyway, it's they just had a recent they're usually doing what they say, decoding the gurus. So it's kind of taking apart people who make not outlandish claims, but sciencey claims, but they kind of take those things apart.
Yeah.
But they recently had a researcher who was very much just talking about how discussions around replicability have changed since over just 10 years, and in a good way. And
yeah, I remember that when people started talking about this, it was people were very defensive about it. I mean, as you might imagine, people were defending their turf. They thought that they, done their due diligence, and then there are these people who say, "Well, look, we have this crisis," and so But I think there's been more acceptance, more recently, about the need to be sort of rigorous in this area.
Yeah, for sure. And there's kind of a second, conversation of neuroimaging people who are doing kind of more statistics at scale, but it's still very similar. It's a lot of overlapping concerns and issues, even though the conversation's sometimes is a bit more technical.
Right. Yeah. I mean, there are a lot of areas of science you could say that there's I mean, the term replication crisis always implies kind of a moral crusade or something. So maybe it's not a crisis, but definitely it's we don't have good community standards for these things. We are beginning to have those, and that's one of the epitaph behind different types of toolboxes and virtual machines and things like that we've talked about with replication neuroscience, where you're building tools for people to more easily standardize their results. So you're not just kind of homebrewing a solution every time you need to do something. You can make sure that you have good control over those things, and I think that improves the results, because it just you go through this pipeline, you're verifying everything, and it's not a huge cost to the labs doing the work.
Yeah.
All right, and like I said, the whole nother kettle of fish are benchmarks in computer science. So that's And I know that like we think that benchmarks are sort of a lot easier than, like we say, well, social science is pretty hard and it's got a lot of interaction there. But like in computer science, surely we can just kind of get these benchmarks and they should be, pretty easy to understand and calculate. But again, you have these problems where you kind of make up your own methods, or you maybe want to see something. There's a sort of confirmation bias that if something is true, you want that to be true, so that's what happens. But again, I think it is like really about, well, it's about community standards, but a lot of times it's also about cost to the researchers. So sometimes these to ensure replicability, it's expensive. It's like you have to spend the time, you have to spend sometimes the money to do these things, and not everyone can. Not every group has the resources. So it just makes it, if you make it easier for them to comply, it's not that hard to do.
Yeah. I would say, I think ARC is an example. The AGI ARC is an example where like our kind of own understanding is also a limitation of the benchmarks that we create.
Yeah.
Like we can't define good intelligence tests for these models when we have trouble creating them for ourselves, or like
right.
creating unbiased benchmarks, or like yeah, you know what I mean.
Yeah. And it's like if we don't understand intelligence very well, we can't make good benchmarks for it.
Yeah. I mean, I see it. I see it in doing all this agentic coding, where like I see it in the issues of testing on synthetic data.
Oh yeah.
Because it's like it can look complex. It can look to the eye, or these other things, but like it's really just as good as your generating function.
Right.
And so if you give it some real data and suddenly everything's off, it's like you've obviously not got something in your synthetic generator.
Right. Yeah, that's a great conversation to kind of continue. We have time for one more thing. So I did want to go over a paper really quickly that is of interest maybe to ecological psychology types, and I think probably Jesse would enjoy this. This is a paper that's now published in this book, Neurocognitive Foundations of Mind. This is a neuroecological architecture for situated cognizing systems. This is Luis Favela, who we've talked about before in Cognition Futures and some other contexts. Let me go over the abstract and then we'll go into the paper. So this is this is a chapter in this book. And so the abstract reads, situated approaches to cognizing, which is defined as distributed, embodied, and extended cognition, or, different ways of doing cognition, basically, have exerted an immense influence on the cognitive and psychological sciences. So distributed cognition is often thought of as different agents doing cognition and then kind of coming together in groups. Embodied cognition, of course, is doing cognition with reference to a body. So the brain or the agent is in a body, and that body conditions how that cognition is expressed. And then extended cognition is extending cognition into the world through artifacts or through the environment. So this is, these are things, of course, that are situated in a context that are hard to separate from their context. So again, when we talk about experimental replicability, if we want to make an experiment like this replicable, we don't need to just replicate the sort of the highly contrived experimental conditions, but we need to put people in a similar environment. So this is the whole sort of debate between very controlled experiments in the lab and these more naturalistic experiments where it may not be as highly controlled, but you're in the right environment, in the right context, and so that's maybe what matters more. Since such accounts typically do not prioritize neurobiology, their impact is felt far less in neuroscience. So when we talk about extended and embodied and distributed cognition, usually we're not thinking about this in terms of neuroscience. We're thinking about this in terms of like behavior, and in cognitive science, it's usually like some behavioral observation where you put people in a airplane cockpit and you look at how they make choices on a control panel, and then you record their responses, and there's no neuroscience necessarily involved in it. So this is where we want to sort of unify ecological psychology with neuroscience and have this neuroecological architecture. The influence in ecological psychology, which stresses the role of ecological information, which is, of course, things like flowing visual cues and other types of dynamical systems type inputs, or things that lead to dynamical systems from the environment. We have this role of ecological information that dynamically unfolds at the scale of organism environment systems that's critical for cognizing, and this is something that is something we need. A substantial criticism of this approach concerns the apparent absence of the brain's contribution to cognition, insofar as other situated approaches remain influenced by ecological psychology. Such criticisms undermine other approaches as well. The primary aim of this work is to present a framework for integrating neuroscience with situated approaches, namely the neuroecological nexus theory, or NEXT. So this is the theory he's proposing here. To be successful, NEXT must both maintain the core principles of ecological psychology and provide testable hypotheses concerning the neurobiological contributions to intelligent organism environment events. The latter is provided by contemporary neuroscience work on the neural manifold hypothesis. In hypothesizing that situated cognition can be explained by interactions among environmental or ecological information and classes of neuronal manifolds, NEXT provides a step towards a more complete understanding of mind, one that spans the cognitive and psychological sciences as well as the neurosciences. So this is something that unifies two different areas. This is the preprint, and so it gets into a little bit of history here. Situated approaches to cognition generally adhere to the view that intelligent systems are always embodied and embedded. So this is something that, again, when we talk about situated cognition, which is usually a sort of a shorthand for these sort of naturalistic experiments, or a theory that we use to interpret naturalistic experiments, we're usually dealing with intelligent systems. It could mean a human operator, or an animal in a naturalistic environment, or it could mean an AI system or an automated system in a body in a naturalistic environment or something like that. So it could be like a robot in a naturalistic environment, or an AI in a naturalistic environment, like an autonomous vehicle or something like that. And so we have to start thinking about these theoretical constructs like embodiment, embeddedness, example. Though there is variety and controversy in how the term is used, as a starting point, embodied refers to the idea that cognition requires a body to occur. So embodied means you need a body. Whatever a body is, And so a body could be like, our bodies, where we have relations between the different parts of our bodies. It could be like a vehicle. So a vehicle has specific movements, it has specific control imperatives, and so those you have to operate within that context. So you have a vehicle that's, say, moving around an environment. It can get to different points by selecting different control mechanisms like steering or acceleration or reversals, and they create different behavioral outputs. And so these behavioral outputs then these different behavioral outputs sort of determine what the intelligent behavior looks like. So if you only have a subset of control imperatives available to you, it doesn't matter what your intelligent behavior is sort of deducing from the environment. The outputs are the important part. And so the outputs might limit significantly what you can actually express as a behavior. So this is embodiment, and this is, of course, important. Their whole conference is an embodiment in, say, like robots and automated systems. But it's easier said than done. There are a lot of open questions in embodiment and how to characterize that vis-a-vis these intelligent controllers. In a more specific sense, this means that there tends to not be a sharp distinction between cognition and non-cognition in the body. And related, the sensory motor processes are important for cognition. So we need to really we can't just think of the body as kind of a conduit for intelligence or for cognition. The body actually interacts with intelligence. So like you acquire information through your eyes or through your hands, and you don't require information, through your stomach. It's not the conduit. But there's an interaction there. So sometimes your visual acuity is controlled by your central nervous system, and there are connections between the body and mind that need to occur here. And this is true of any kind of a controller with any kind of body. So it could be like a autonomous vehicle controller. It has to have interactions with the vehicle, and in fact we have that in autonomous vehicles with sensor systems across the vehicle. In a more specific sense, this means there tends not to be a sharp distinction between cognition and non-cognition. And related, the sensory motor processes are important for cognition. There is a spectrum in how weak or strong these positions are. At the weaker end of the spectrum, cognition is primarily a brain event, but the brain areas associated with the body's sensory motor processes play important roles in causing and enabling even higher forms of cognition, such as abstract reasoning and language. At the stronger end of the spectrum, cognition is not primarily a brain event, with the body's non-neural structures and processes causing and constituting even those previously mentioned higher forms of cognition. So this is like where we talk about weak and strong embodiment. Weak embodiment is where we kind of think of the brain as being within a body, yes, but the brain is doing most of the work, and the body is just there as sort of an instrument of the brain. In strong embodiment, the body is actually doing most of the computation, or it's balancing out with the brain or the controller. So, this is like a debate within embodiment that's important here because, you have to deal with that, the role of the body, basically. So this is where we think about embodiment, and it's important to kind of be very specific what we mean by embodiment. Okay, so, and then we have to go back, when we think about ecological psychology, we need to go back to behaviorism and think about sort of our route out of behaviorism. So when we kind of came from behaviorism and moved into the cognitive revolution, we were kind of left with some intellectual baggage. Some of that is where, even from the earliest days of psychology and behaviorism, there was some form of functionalism, which means that like the way we thought of all mental processes was primarily functional, meaning that something did something else. So when we think about these interactions between body and brain or body and controller, we're always thinking in terms of functionalism. So it's like, what is, or why does this behavior exist? It must exist for a reason. It's probably to adapt to its environment, to allow the organism to adapt to its environment. And so this is, of course, both good and bad in a lot of ways, because sometimes functionalism can constrain us, and sometimes it serves as a useful explanatory device. So this kind of found its way through behaviorism, and then it found its way sort of into the cognitive revolution as well. And so, but in behaviorism specifically, it had three main commitments. The first is that psychology is the science of behavior, and that the sources of behavior are basically external, which means that they're in the environment. And that when you talk about mental concepts, you shouldn't, and rather you should talk about behavioral concepts. And this actually is one of the differences between cognition, or the cognitive, what would become cognitive science, and ecological psychology, is that ecological psychology sort of retained this aspect of the source of behavior, which is that it's external in the environment instead of internal to the mind. And so this is kind of describing this distinction between weak and strong embodiment. And so then, we kind of talk a little bit about the cognitive revolution going through this history, and then, of course, the position of Gibsonian ecological psychology within that. And, of course, sort of Gibsonian ecological psychology bridges this gap between perception and action and sort of behavior, or some of the things we think about in cognition, and behaviorism and some of the features of behaviorism. And there have been a lot of applications of ecological psychology to development, to human-machine interaction, to virtual reality, and etc. And so then again, in ecological psychology, we think of perception as being direct, meaning that we're really kind of drawing from ecological information, and we're building our models based on that. The information is out in the environment and not necessarily originating in the brain or in the mind. And so, we have different ways of, dealing with perception. That's, where we treat perception as direct. It doesn't rely on any sort of intermediate representation. And then, again, okay. Then second, perception and action are continuous. So again, this is where, in a lot of psychology experiments, we present these discrete trials, and we repeat stimulus presentation over and over again. In an ecological setting, perception and action are actually continuous. They flow, and the order of those stimuli are very important in terms of acquiring that information. So you can't just present things in these discrete, discontinuous trials. They have to be in this order, in this continuous flow. And so this is actually quite a big challenge to a lot of experimental designs, because if we have, like, say, a naturalistic design, we might put an agent out into the environment and let them interact with the environment. We get this ecological information, this continuous information. We get this perception-action loop that forms. But that action is quite unique to that set of interactions. So as you can see, this is very hard to square away with replication, because every time you run the experiment, the flow of information from the environment will be somewhat different. I mean, even if you have the same environment, a highly controlled environment, it will be somewhat different. But this is necessary to understand the true nature of a perception action loop, and it kind of speaks to how maybe, the way we think about doing experiments does not square away with the needs to understand how the mind works or how behavior works. And then third, we talk about affordances. And so as a consequence of perception being direct and the continuity of perception and action, it follows that detected ecological information can specify meaningful opportunities for action, namely affordances. In short, affordances are perceivable opportunities for behavior, so they're just features within an environment or somewhere where you kind of latch onto them. So they're kind of like intuitive points where you can kind of pick up. So if you have, like, say, for example, a door, if you see a handle to the door, you learn that that handle is good for either pushing or pulling the door on a hinge. So you learn this in development. You interact with the door. And once you interact with the door, you get an intuition for how it operates. And so any door that you encounter, you try to push or pull it with the handle. Now, if a door maybe slides along the track and you try to do that, then you have problems generalizing. But if a door always behaves the same way, then you just simply use that intuition to guide the action, and you can successfully generalize that information. So Gibson talked a lot about affordances. They're both objective and subjective properties of an object. And this is, it's sort of a unique way to look at generalization in training and intelligence. So if you have an agent that needs to generalize something, or some pattern in the environment, if it's an action type of a pattern, like if a robot is trying to move a door or throw a ball or do something with something, it needs to actually go through the action, try out different things, and build this intuition of what this sort of thing does, and then it can apply that more broadly to other classes of similar thing, and that's how it actually would learn. It's not going to learn from presenting all possible options as to how this thing is used, because first of all, it can't necessarily select from those options, and secondly, you can't really test those out in the environment that you're in. It's going to be very hard to do that. So this is why a lot of robots have problems with tasks that seem effortless to us, because we've sort of automated, we have these sort of automated or procedural behaviors that we don't think about how we execute it. We just kind of learn the sort of the procedure and we internalize that. Okay, so fourth, psychology is about the organism environment system. So we have to have the system of both the organism and the environment. This is this extended cognition where things are offloaded to the environment, where things are, we extend our cognition into the environment. And we use the environment and our mental models interchangeably. Okay, so this just kind of goes through this history and talks about sort of situatedness. And then it sets up his neuroecological cognitive architecture. So this is the neuroecological nexus theory. And so next is appropriate for the investigation of cognizing organism environment systems that have nervous systems. Since cognitive systems are always situated, next is committed to the position that such systems necessarily include organisms, which are embodied, of course, that are tightly coupled with or constituted by their ecologies. That's embeddedness. So that, you have organisms that have a body, but they're tightly embedded in these ecologies or these environments. The nexus part refers to both the framework as a point of convergence for ecological psychology and neuroscience, and as a nexus that constitutes the primary causal and or constitutive links or junctions that connect the various elements of a cognizing organism environment system. So next is defined by 5 hypotheses. Hypothesis 1 is that the organism environment system is the privileged spatiotemporal scale to understand cognition. So we don't care about like the details of the brain, we care about the organism and the environment. But we also care about this multi-scale nature of the problem. So sometimes we want to drill down to neuronal activity and we want to have this information, but we want to link ecological psychology in the environment with neuronal activity in the brain. And so we need to establish this sort of organism environment system, but then have the access to these more detailed scales in the brain. Hypothesis 2 is that neural population dynamics generate the relevant states for cognizing. So this is where we have these population dynamics that are at a lower level of organization. And so we, instead of sort of thinking about cells as just neurons, we need to think about neural populations. And so a lot of times in cognition, neural populations are the things, the scale of interest, where neurons work together to produce representations, and their activity will, tune those representations accordingly. So there's a lot of theory and experiment behind that. A neural population is a neuronal group that has been selected for over species development timescales. So you have specific populations of cells in the visual cortex that are tuned to extract things like columns, stripes, blobs, and then that feeds into higher level representations of images. You also have populations in sensory motor cortex that are tuned to different limb movements, directions of limb movements, things like that. Hypothesis 3 is that cognizing is based on low-dimensional neural dynamics. So this is where this manifold comes in, and manifolds are sort of the current theory of how neuronal signals are organized. The thinking is that there's this topology, or this representation that forms a topology. So you have these high-dimensional structures and low-dimensional manifolds, and so there's a mapping between the two that's what we call homeomorphic, which means that they share the same structure even though they look very different. So that's kind of a tool we can use to understand representations of large conceptual spaces in low-dimensional structures. All right, and then of course it gets into a lot of things about the neural manifold hypothesis and how things that, say, this rat is interacting with the environment, moving around the environment, are represented in the brain as these neural manifolds, and then this was represented by a topological structure like a torus. And so you would have like these movements and these interactions encoded as the shape, and then you explore that area of the shape through neuronal activity, and everything kind of maps to this torus, which is the representation of the environment. So then this is a hypothesis 4 is that the body organizes into low-dimensional synergies. So this is going back to synergetics from Kelso, which is where you have limbs that sort of are controlled together into groups, muscles that are controlled together into these synergetic groups, and these groups then act as sort of outputs for the neural manifolds. So a lot of times you can take a space and decompose it into a manifold itself. So there's this uncontrolled or unconstrained manifold hypothesis that basically breaks the environment down into a configuration space. If you have, like say, a workspace in front of you like a desk, how do you have to move your limbs to reach every part of that space? And there actually are quite a few options if you were to consider every possible movement as an individual degree of freedom. But of course, our limbs don't operate like that. Our limbs operate as sort of these grouped muscle synergies, these grouped limb synergies. And so our limbs move in certain directions, and we have access to maybe a subset of movements to reach every part of that space. So when we move our arm, we don't, bend our wrist and bend our arm the other direction, or our elbow the other direction. We actually kind of, everything moves in a coordinated way so we can reach different parts of that space. And so there are a lot of different mathematical tools that allow us to decompose these spaces and suggest optimal placement of objects and things like that. And of course, this holds true for most things in the environment where, the brain actually computes sort of the most efficient path to a target. And it's a really interesting dynamical systems theory underlying this. And then hypothesis 5 is that cognizing fundamentally emerges at low dimensional states of the organism. And so next claims that cognizing is a multi-scale phenomena that emerges from systematic relationships among brain, body, and the world. So each spatiotemporal scale is high dimensional, but the principal structure of each is low dimensional. Okay, so this is something where, we're working from these neural populations, we're working from these uncontrolled manifolds that describe body synergies acting in the world, and then we can from there build a model of action in the world from neural populations. And so this is an example here of what a rodent in a burrow, and it's trying to navigate its way out of the burrow, and it's building this representation of the environment, and it's then acting upon it, and it's producing these muscle synergies that allow it to reach different points in the environment. And it has this model of an unconstrained manifold or an uncontrolled manifold that tell it kind of the best places to go for things. It's a nice paper because it stitches together a lot of ideas into what is essentially a cognitive architecture. There isn't a formal architecture program that he's producing here, but I think you can see the logic in a lot of this. Okay, so that's that paper. All right, so I think that's all for today. Thank you for attending.
Thank you, Bradley, for hosting this meeting.
Yeah.